So the chapter 4, that is principles of inheritance and variation. Okay, so like Mendel's laws of inheritance, right? Now, seven pairs of contrasting traits in pea plants studied by Mendel. Seven pairs of contrasting traits, and Mendel conducted with such artificial pollination, that is cross pollination experiments, using several true breeding pea lines. A true a true breeding a true breeding line is one that, having undergone continuous self-pollination, right, shows the stable trait inheritance and expression for several generations. So Mendel selected like 40 true breeding pea plant varieties as pairs, which were similar except for one character with contrasting traits. Okay, so some of the contrasting traits selected were like smooth or wrinkled seeds, yellow or green seeds, or inflated, that is full, or constricted green, or constricted, that is constricted, then green or yellow pods, then tall or then tall or uh, then tall or then tall or dwarf plants. Okay, so the contrasting traits studied by Mendel in P is characters, that is stem, that is tall or dwarf, then flower color, that is violet and white, right? Flower position, that is axial and terminal. So axial is the dominant pod. Okay, and then pod shape, pod shape, that is inflated, that is full and constricted. Okay, pod color, that is green and yellow. A sheet shape, that is round and wrinkle, and sheet color, that is yellow and green, right? So contrasting states studied by Mendel in P, right? So this is it, like um, uh, now, like in here, like artificial selection and domestication from ancestral wild cows, we have known, that is Indian breeds, that is Sahiwal cow in Punjab. Okay, and the John Mendel conducted hybridization experiments on garden peas for like 7 years, that is 1856 to 1863. Okay, so 1856 to 1863, and proposed the laws of inheritance in living organisms. Okay, and this is it, like now inheritance of one gene, we get the F1, then F2 after selfing, uh, and like removal of anthers is emasculation, it is female parent and male parent, that is bisexual flowers. Okay, the Mendel then self pollinated the tall F1 plants, and to his surprise found that uh, some of the offsprings were dark. The character that was not seen in the F1 generation was now expressed. The proportion of plants that were dwarf were 1 by 4th of the F2 plants, while 3 by 4th of the F2 plants were tall. Okay, so the tall and dwarf traits were identical to their parental type and did not show any blending. Okay, and that is all the offspring were either tall or dwarf. None were of in between height. Okay, now uh, okay, the similar results were obtained with the another traits that he studied. Okay, only one of the parental traits was expressed in the F1 generation, while at the F2 both the traits were expressed in the proportion 3:1. The contrasting traits did not show any blending at either like F1 or F2 stage. Okay, so he named it like the factors which control these characters, right? Now we call it as genes, and genes, these are the units of inheritance. Now they contain the information, and genes which code for a pair of contrasting traits, which genes which code for pair of contrasting traits are known as alleles. So they are slightly different forms of the same genes. Okay, now you see, there is dominant allele, then recessive allele. So the monohybrid cross, the capital T small t plant is heterozygous for genes controlling one character height. So only one character height, it is monohybrid cross. If it was two, then it is called dihybrid cross. Now there is segregation, heterozygous, and heterozygous, and then heterozygous, and then that is Punnett square. Okay, so the production of gametes by the parents, the formation of the zygotes, the F1 and F2 plants can be understood from a diagram, Punnett square. It was developed by a British geneticist Reginald C. Punnett, and it is a graphical representation to calculate the probability of all possible genotypes and phenotypes of genotypes, but it also tells the phenotypes as well of offspring in a genetic cross. Okay, you can see the monohybrid cross phenotypic ratio is tall is to like dwarf, that is 3 is to 1. Then genotypic ratio capital T like capital T small t and small t, that is 1 is to 2 is to 1. Okay, a Punnett square used to understand a typical monohybrid cross conducted by Mendel between true breeding tall plants and true breeding dwarf plants. Okay, true breeding tall plants and true breeding dwarf plants. Okay, so this is it. Then so this is it like, and uh, so this is it. Okay, uh, then there is like test cross. Now what is test cross? So when we get like so that is, for example, whether a tall plant from F1 or F2 has like capital T or capital T small t composition cannot be predicted. Therefore cannot be predicted. Therefore to determine the genotype of a tall plant at F2, therefore to determine the genotype of a tall plant at F2 to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from the F2 with a dwarf plant. Okay, this is so he called it as a test cross. So in a typical test cross, an organism like pea plants here showing a dominant phenotype and whose genotype is to be determined, that is F1, we are calling it like the hybrid one, is cross. It can be homozygous or it can be heterozygous. So is crossed with the recessive plant instead of self crossing. The progenies of such cross can easily be analyzed. Okay, to predict the genotype of the test organism. Now you see, mm, okay, so unknown flower is homozygous dominant. You see, when the hybrid is like the homozygous, the unknown flower is homozygous when all the flowers are violet. Okay, it is violet, and 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 in the second case, when the unknown flower is heterozygous, the half of the flowers are violet and half of the flowers are white. So this is it like capital W small t, and so dominant phenotype. So based on his observation on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today these rules are called the principles or laws of inheritance. The first law or law of dominance, and the second law or law of segregation. So law of dominance. Okay, the characters are controlled by discrete units called the factors, and these factors are occur in pairs. Okay, so in a dissimilar pair of factors, one member of the pair dominates, that is dominant, the other, that is recessive. So law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 and the expression of both in the F2, that is tall and dwarf. So it also explains like the proportion of 3 is to 1 obtained at the F2. Okay, so why so the proportion of the 3 is to 1 is explained by the monohybrid cross. Okay, 3 is to 1, right? Remember the 3 is to 1. It also explained at the like F2. Okay, so now the law of segregation. This law is based on the fact that the alleles do not show any blending and that both the characters are recorded as such in the F2 generation, though one of these is not shown at the F1 stage, right? So this is it like, now the incomplete dominance. So you see like red and white is crossed and all pink flowers are formed, and the phenotypic ratio is also 1 is to 2 is to 1 and genotypic as 1 is to 2 is to 1. So it is like the snapdragon, that is results of monohybrid cross in plant like snapdragon. So where one allele is incompletely dominant over the other allele. Okay, so incomplete. Now what is incomplete dominance? There was like dominance when one in a dissimilar pair of alleles, one is dominant over the other. So but in case of incomplete dominance, so when experiments on peas were repeated using, we found that F1 had a phenotype that did not resemble either of the, okay, so F1 had a phenotype that did not resemble either of the two parents and was in between them. So was in between them, like they both alleles are showing their partially characters. So the inheritance of flower color in the dog flower, that is snapdragon or anti-remnant specium. uh, okay, so the inheritance of the flower color in the dog flower, that is snapdragon or antirrhinum sapien, in a cross between like true breeding red flower, that's capital R, and true breeding white flower, that is white flowers, that is a small, the f1 capital R small r was pink. So it was like pink, and when the f1 was self pollinated, the f2 resulted in the following ratio, that is red is to pink is to white. So red was like 1 capital R, and like 2, that is capital R small r, that is pink, and 1 is the white. So okay, so this is why like here the genotype ratios were exactly as we would accept in any Mendelian monohybrid cross, but the phenotype ratios had changed from like the 3 is to 1 dominant recessive to, I mean, 1 is to 2 is to 1. Okay, so this is it like, and the reason for that is like, as you can see, the normal less efficient enzyme or non-functional or no enzyme at all. um, okay, the phenotype trait will only be dependent on the functioning of the unmodified allele. Okay, unmodified allele, and modified allele can be like non-functional enzyme or no enzyme at all. Okay, so codominance. So what is codominance? So where F1 resemble either of the two parents, that is dominance, or was in between, that is incomplete dominance. So if it resemble either of the two parents, that is dominance. If it resemble either of the two parents, then it is dominant. then 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 it is dominance. But if it is present in between, that is incomplete dominance. So was in between, that is incomplete dominance, and resemble either of the two parents, that is dominance. So but in codominance, like the f1 generation resembles both the parents, but in case of codominance of the f1 generation resembles both the parents. Okay, now ABO is an example of codominance also. It is an example of multiple allelism. Okay, and law of dominance. Okay, and ABO blood group is controlled by gene R. Now it has three alleles, that is IA, IB, and R. Now IA and IB. Now ABO blood group is based on like presence or absence of the antibodies and antigen present on like in RBC, or like maybe in the plasma. So no, the production of like antigen and antibody. So this is it. Now poor I allele does not produce any kind of the sugar. So and we are diploid. So we get only two of the three alleles. But this is the multiple allelism, so three alleles are present. Okay, now this is it. Like when I and IB are completely dominant, so AB blood group. Now so this is it now. Okay, so when I and IV are present together, they both express their own types of sugars. So this is because of the codominance, right? Okay, so here blood cells have both A and B types of sugars, right? So three different alleles, and six different combinations of these three alleles that are possible. So six different genotypes of the human ABO group. So you can see the six different genotypes. So IA, that is A, I, B, AB, AB. So IA and small I, that is A, I, B, AB. Okay, then IB, that is B, and IB and I, that is B, and I, that is O. So as you can see, 1 2 3 4 5 6, so six different genotypes. And like the three I mean the four A B B and O. So these are like four different types of as you can see, like the phenotypes. Okay, so this is an example. So ABO blood grouping also provides a good example of the multiple allelism, right? Good example of multiple allelism. So this is it, like. And so when we study the population studies, so multiple allelism can be found only when the population studies are made. So this is it. So a single gene product may produce more than one effect. So star synthesis, an example of pleiotropism. Okay, uh, a single gene product. Now product, we are talking about product. So single gene can affect control multiple characters. So and single gene product, I mean gene product, may we are talking about the product. Okay, the now that product controlling the I mean giving the like more than one effect. So this is also pleiotropism. Okay, so star synthesis, like capital B capital D, it is homozygous, it is round. Now it is large, it is round, and when it is mature, it is large, right? So small b, it is wrinkle, right? It is wrinkle, and when it is mature, it is shrink. That is small in size. But when capital B and small b, it is of intermediate size. So when it matures, like so when it matures, like you will see, like it's like in between the size. So this is it, like so if like starch gain is considered as a phenotype, then from this angle the alleles show incomplete dominance. So you see, capital B, small b. So this is so no fucking